Okay, so cytokines, receptors, and signaling. We kind of, we covered kind of, the receptors, and for T-cell receptors, B-cell receptors, and that you have these signaling events that go through these cascades, and they activate multiple transcription factors. But now we're going to cover kind of how kind of another event that is important, and probably one of the most important things for the immune system is cytokines, because the cytokines, you have your B cells that are going to see antigen, or your T cells that are going to see antigen, but it's the cytokines that are going to tell them what to do about that signal. And so there's a second signal, and almost always going to be a second signal you receive from cytokines. So cytokines are these small, soluble molecules. Usually, sometimes they're even membrane-bound molecules, but those can also be released. And so these are going to be kind of the regulators of the immune response. They can tell cells to start proliferating, they can tell cells to die, they can tell cells to start moving. So it's basically how our immune cells are going to communicate with each other. So general properties of cytokines, what do we got? Proteins, they're small proteins that mediate the effector function. They can act in different ways. So we always want to break this down into kind of how are they going to function? Are they going to function systemically? Are they going to function locally? Or are they going to function very locally in an autocrine fashion? So we have our endocrine. So if a cytokine is secreted by a cell and can act over a long distance, even passing through the circulation, then this is going to be called endocrine action, or it's going to have an endocrine action. And some cytokines have endocrine action. They also might have paracrine action or even autocrine action. So more than one cytokine can have more than one effect. Paracrine action is when that cytokine is communicating with the cell that's angstroms away, just very close. And then autocrine action is when a cell secretes a cytokine and it binds to its own receptor, so it's kind of talking to itself. It's going to respond to its own set of cytokines to become activated. And IL-2 could be one of those. IL-2 is going to, for example, a T cell becomes activated, it starts secreting IL-2, and then it has a low affinity receptor, but because it's pumping out IL-2, right next to it is this low affinity receptor, some IL-2 binds to that, and we'll talk about that, and that actually will cause a signaling event, and then it'll actually upregulate another receptor portion of the receptor for IL-2, make the high affinity IL-2. So it's this is, and IL-2 can have also distant effects, systemic effects. So
IL-2 goes endocrine and outocrine?
Mm-hmm. Yeah, and in fact, we'll talk about it for just a little bit. Some uses of IL-2, for example. It's one of the more important. Not that any aren't important. It's just one of the more studied, I guess, cytokines. We've known about it for a long time. It was the first cytokine that we made a recombinant for. So most cytokines are soluble proteins released by a cell. They bind to a receptor on another cell or themselves, and they induce this biological effect. But some, especially the TNF family, can be membrane bound and can be released as well. So again, their functions can be when they bind and signal, they can change the differentiation state of the cell, the proliferative state of the cell. They can induce survival or death of that cell. And again, I'm going to use IL-2 as the example. If I take a bunch of T cells out of somebody and I put them in a dish, unless I give them certain signals, they're just going to die. But if I give them enough IL-2, they're going to proliferate, they're going to survive, There are certain other things that you'll have to give them, and a lot of times there are multiple cytokines that need to give a signal for them to really become activated and proliferate and survive. And there are other cytokines that might give them a signal to die. So, and sometimes the same cytokine, depending on concentration, maybe how long it's binding, the combination of cytokines, it might induce one signal in one situation and a different signal in a different situation. We'll talk about the TNF, how there are some of those that are important in inflammation and inducing an immune response, but they also can induce apoptosis. So we have cytokines associated with innate immunity, IL-1, TNF-alpha. Most of these are associated with inflammation. So those first cells that come in and start secreting these cytokines are they're causing local inflammation. They're causing blood vessels to allow more cells into the area. They can induce IL-1 can induce if it gets into the bloodstream, it can induce fever. So things like that. You've got your adaptive immune cytokines, IL-2, IL-4. So IL-2 is well known for inducing helper cells will secrete IL-2 and become these Th1 cells that are inducing a cellular response, whereas IL-4 is going to induce more of a humoral response, and IL-17 is a newer, one of the newer, we're starting to understand more and more about IL-17 is more of this kind of almost allergic or inflammatory responder. And in some cases that might be good, and in some cases it might be bad. Secreted, and these are secreted mainly by T and B cells. And then these innate cytokines are secreted by our more innate macrophages, neutrophils. And you don't have to memorize this figure, but it is just to highlight that there's redundancy, there's what's called pleiotropy, and so there's a lot of cytokines that are secreted by different cells. Some of these cytokines are secreted by multiple different immune cells, and there's a lot of crosstalk, and some cytokines will inhibit a certain response from another cytokine. So there's a lot of pathways, and we're still learning about these, and we're researchers are still. This is one of those areas where we're still learning a lot, and it's a lot more complicated than you think. So we have our interleukins, these like IL-1 again, inflammatory response, which can be good, but then chronically speaking, it can actually be bad. And so, for example, they found that antagonists of IL-1 can actually help treat cancer. And so you may want IL-1 to induce a response against something initially, but then long-term IL-1 might not be, might induce tolerance in a situation where you may not want it. You have TNF or tumor necrosis factor, alphas. These are other inflammatory responses. Interleukins 12 and 6, and you don't have to memorize this. This is just highlighting some of the cytokines in these immune responses to highlight how kind of complex and how many different players, different cells will secrete different cytokines or have receptors for different cytokines. Or we have all this epithelial cells might have receptors for TNF, but whereas our immune cells have lower affinity receptors for TNF, so that they're less, they're kind of less affected by TNF. So cytokines can mediate activation, proliferation, differentiation, as we said, and the effects can be, and so these are the terms, I guess, that I should have highlighted that we need to know. Pleiotropic, where one cytokine can produce multiple effects. it can affect different cells. It can have more than one response. So there's this playtropy. So depending on the context, there's redundancy. A lot of times there are more than one cytokine that will do the same thing. IL-2, I think IL-15 will bind to the same receptor. So there's multiple cytokines where they all have the same effect, but there's this kind of redundancy built into the system to make sure that we have a proper response. There's synergy where two or more cytokines work together to induce the same effect. And then antagonism is where one cytokine will drive one response, the other will drive another, and they may be competing, or they may even compete for the same receptor. They might compete, or they might send different signals, and one signal blocks an enzyme that is downstream of the signal of another one. So there's antagonism between some of these cytokines as well. Another area where that is helper T cells can become Th1 cells, or they can become Th2 cells, and it depends on the cytokine milieu that's around when they become activated. So they have to make that decision. Do I become a Th1 cell that I'm going to help this cellular response, or am I going to become a Th2 cell where I induce a humoral response? And those are going to be based on, do they see IL-2 or are they going to see IL-4, and those are going to compete for signaling.
So which one is the helper T becoming cellular?
So a Th1.
Yeah.
So what cytokines would drive it? Interferon gamma, IL-2 would drive a Th1 type response, whereas like IL-4, I think 8610 will drive more of a humoral response.
So that's a Th1 is IL-2 and
IL-2 and interferon are classic inducers of a Th1 response. Yeah. So here's just a graphic representation of what I was talking about, playtropy. IL-4, it will activate proliferation in B cells and T cells and mast cells. So it has the same effect on multiple cells because they all have receptors for it, and it's going to affect a broad range of immune cells as opposed to being very specific. There's redundancy, and you say IL-2, IL-4. These all, even though they may induce a different subset, they all are going, in T cells, in B cells, they're all going to induce proliferation. So they might have a different effect on T cells, but here on B cells, there's this redundancy. IL-2, IL-4, IL-5, they're all going to produce proliferation in B cells. Synergy, IL-4 and IL-5 can work together to induce class switching, and so they will have this synergistic effect. Whereas antagonism, and we're going to use IL-4 and interferon gamma here, we're looking at a B cell, but the same thing could be said in maybe T cells. So in B cells, IL-4 will drive class switching to IgE, but interferon gamma is going to block that. And so interferon gamma and IL-4 are kind of competing also in T cells for T cell differentiation to become a Th1 or a Th2 cell. Yeah?
What is class switch?
So class switching, we're going to cover that in when we talk about B cell receptors, but it's basically there are different constant regions in the receptor, the B cell receptor, and so it's going to stop making one and make a different one, and that could change whether that receptor is like membrane bound versus secreted. And some are involved more in allergies than others, and so that class switching, it's still the same specificity for whatever its antigen is, but the rest of it's a little bit different, and so it could be secreted versus membrane bound. It changes kind of how it behaves, I guess. So whether that B cell is going to go around looking for antigen and then engulf it, as opposed to just start kicking out antibodies, would be a class switching event. And so it makes sense that IL-4 and interferon gamma are kind of focusing, these are kind of two cytokines that are kind of driving a humoral response versus maybe more cell-based response. So interferon gamma it's inducing this more cellular attack where it wants cells to specifically come and target something. Whereas IL-4 might be, hey, there's a parasite. We're not, like interferon gamma would be an antiviral responder, whereas IL-4 might be more parasitic, something outside of the cell. So you want to attack it differently. Your interferon gamma is going to induce this cell. We're going to start killing cells that are infected. Whereas IL-4 is going to induce more of this, hey, let's send out all these antibodies and make sure that we're finding whatever's floating around in the periphery and kill it. So it's kind of two different responses, and that's why we've developed kind of this antagonism between, because there's different responses that we want to have for different pathogens. Okay. So then we break cytokines down into six more families, I guess we're going to call them, based on structure and a little bit of function. We have our interleukin-1 families, and some of the earliest cytokines, they call them interleukins because they communicated between leukins or white blood cells. And these are your inflammatory cytokines like IL-1 alpha, beta, IL-1 receptor alpha, and IL-18, IL-33. So most of these are involved in inflammation. So we have our class 1 cytokine families, IL-2. So these are small cytokines. These are just very small cytokines, but there's a huge array of different cytokines in this family. IL-2, IL-3, IL-4, IL-6. So a lot of these are involved. Some of these are involved in humoral responses, some in cellular responses, some in inflammation, GM-CSF, GCSF, some in cell survival, cell growth, and so we are prolactin. So a lot of these are involved in a pretty broad range of effects, but they're all pretty conserved in their shape and size. We have our class 2, interferon class 2 cytokines. Our interferons, interferon gamma is one of the most, interferon gamma. So these interferons are basically called that because when they first started growing cells and they came in contact with virus, and then they took supernatants from those cells that didn't have any cellular components. They had some component that was secreted that had direct antiviral effects, and so they called them interferons. And so that's this class of interferons. They have kind of this antiviral effect without having to have an immune cell present. They have receptors on other cells, and they'll induce events that help to inhibit viral growth. It's good to know the different classes and maybe a few of the key antibodies, like IL-1 beta would be my favorite. In this one, so in the study guide, I'll try and also say if I'm gonna have questions on these, and I'll kind of tell you, hey, focus on, I want you to know that IL-1 beta is part of the interleukin 1 family, or here IL-2 would be one of my favorite cytokines. Interferon gamma is a really huge player. TNF alpha, CD40 ligand, so this is involved. So now in our tumor necrosis factor family, these are highly involved in cell survival and cell death. CD40 ligand is going to be this co-stimulatory molecule ligand, so it's going to be the signal. If it's signaled through this ligand, then this cell is going to go ahead and survive. If it doesn't get that signal, it might die. So here's where, depending on if it gets this signal when it's activated, when it sees antigen, it might survive. If it doesn't, it might die. Whereas fast and fast ligand, these are always going to induce apoptosis when fast and fast ligand bind together. And tumors, for example, will put out like little mines of fast that binds the fast ligand and kills immune cells as they're coming in. And in nature, what you say, well, why did we evolve these if they're just going to kill immune cells? It's because if we have an inflammatory response and we're starting to even damage our own cells, we have to have a mechanism to kind of shut that response down. Otherwise, we'll basically have autoimmune-induced this cascade event and basically melt ourselves. So if we didn't have these molecules to shut down our immune cells, they would go on out of control. But then things like tumor cells that hijacked these factors. And so you'll find that in studies where they look at microenvironments of tumors will have lots of these factors to kind of kill the immune cells that are in that environment. The interleukin 17 family, this is recently discovered, so this has probably only been in the most recent editions of this, involved in inflammation, but also in a lot of allergic responses. And people have taken this and said, hey, if these are normally involved in autoimmune responses or responses to self, maybe we can use this to our advantage and try and target cancer cells with these type of cytokines and overcome tolerance. So people are studying really in detail this interleukin 17 family. And then we have our chemokines, which are a subclass of cytokines. So they're also cytokines, but they're involved in motility of cells and basically creating a gradient that cells can follow to an area, or telling cells to start moving to the lymph nodes or move into the periphery. So these are really important as well. Or they could be involved in bringing cells to an area of inflammation, or bringing in dendritic cells. So IL-1 family promotes inflammation. Again, that's the key factor to remember here. What the book doesn't talk about is long-term IL-1 can cause chronic effects, and it can have negative effects.
What do you mean chronic effect?
So if you have chronic secretion of, so if IL-1, you have IL-1 cells that are secreting IL-1, it's an acute event. They're going to bring in cells. Those cells are going to start damaging tissue and reorganizing tissue. They're involved in tissue remodeling and bringing in white blood cells, macrophages, neutrophils. So IL-1 is really important. But if it's secreted chronically, it's actually can be involved in tissue remodeling, tissue damage. Tumors, for example, they found that if we block IL-1, if you block IL-1 with an antibody, you can actually affect tumor growth, because the tumors are making their own vasculature, and they're creating this environment where they're remodeling the whole environment in their surroundings. So
this can be anti-cancer?
if you block it, it could be a cancer treatment. And there are some cancer treatments where they've focused on blocking IL-1, for example. They haven't by themselves been very effective, but
It's not bad.
it works great in mouse models.
Oh, so the absence of IL-1 helps cancer treatment?
Yes. So, like for example, there's an IL-1 receptor that does not signal, and so it will actually. There's two receptors. There's a class 1 receptor that will signal through IL-1, and there's a class 2 receptor that actually doesn't have the signaling mechanism on the tail end side. And so that antagonist, they call IL-1 receptor antagonist, it can be soluble, you can treat people with it, it basically will soak up any IL-1, it'll bind to that receptor, but it won't signal through cells, and they've used that to treat cancer.
So it's like a non-receptor will take protein,
It competes with the receptor, but it doesn't signal, so, and that's,
They're not proliferate the
yeah.
cells.
Yeah, it doesn't, cells don't proliferate, you quell the immune response, and tissue remodeling stops, which is detrimental to tumors because they're going to, a lot of times they need vasculature, they need nutrients to continue to grow. Okay, so
So the cancer cells are the ones producing the same amount of interleukin-1?
a lot of times, or they'll hijack immune cells. Okay. So, IL-1 alpha and beta are one class. They bind to this IL-1 receptor, or the receptor antagonist. They have this inhibiting ligand receptor pair to shut down. So this is this IL-1 receptor alpha, or receptor antagonist, I'm sorry. IL-1 receptor antagonist, it binds to the IL-1 receptor and prevents it from binding with its function. So you've got that, but then also another class. My computer just shut down or something, but it's going. IL-18 is another similar. It's related to. It's another one of these IL-1 families, and you can inhibit it through binding of cytokine before it binds to the receptor. So there's different ways to block it, either block the receptor itself, and these are natural events that will happen. So cells will, immune cells will can secrete these antagonists or not. So it's not something you need to memorize. It's just good to know that it can happen. And then IL-33 is another member of that family, and it has also an inhibitory receptor shutting down that. So each one has receptors that they can signal through, or either receptors that will block or compete for the cytokine, or be secreted and bind to the cytokine, keeping it from binding the cells. So we have to have this tight control over, especially these inflammatory cytokines.
If you have a cytokine IL-1, that means they promote inflammation.
Yes.
So there are some receptors exist in the body. They bind the
Yes.
IL-1.
Yeah.
So
Then we also have, these cells can also produce antagonists, or immune cells can shut down that response over time by secreting antagonists. So a lot of these responses, for example, IL-2, once you have a little bit of IL-2, it's basically this cascade effect that it will now secrete more IL-2, and you get this huge effect. And you want it to happen very quickly, so you want immune responses to happen quickly, but then you need ways basically to soak up all that cytokine to shut down that response. And so these classically, these inflammatory cytokines will always have some type of antagonist or decoy receptor to compete out and shut down that response.
but how do they know when they helping inflammation?
Sometimes it's a timing thing. Sometimes cells will, after a certain amount of time, will start to secrete the suppressive, the receptor that is suppressive. Sometimes it's a timing event, and so you'll have one receptor that will bind, and it will, over time, it'll eventually start to signal for the other receptor that's going to. So you might have an acute event, but then you start to suppress that inflammation over time. And that happens in T cells as well. When we specifically focus on T cell signaling, you'll see that there are certain receptors that are expressed early on in T cell activation, and then other receptors will eventually get upregulated that have the opposite effect. So you'll get these T cells will start proliferating like crazy. They'll start attacking things, but then the same signals that turn them on will actually start to suppress them. And so you can kind of create an event that you know is going to be very damaging, but you want that to happen, and then you want to start shutting it down. So.
I think it must be a mechanism then to alert them to do something about it, what they use, right? You know, first they makes it come back.
Yeah. So, but for this, I just want you guys to know what the different classes of cytokines are, kind of what are some of the family members in them, and what are some of the key things involved in those. And we're going to be discussing these specific cytokines when we talk about T-cell activation, when we talk about B-cell activation. So we're going to focus in more on these pathways. So definitely we'll be talking more about them and their effects, whether positive or negative, on immune cells and what those are. But right now we're just going to focus on just trying to get through what are the different classes and what are the cytokines in those classes and key things. Signaling, again, we just like we had in T cells, you have this cascade of, here we have MID88, which is an easy one to remember, and it will recruit, engages receptor, this basically binds, recruits these IRACs, and they phosphorylate and trap 6, and then you get, so basically it's the same type of pathway. I don't expect you to memorize this pathway per se. One pathway that's important to memorize, I'll point it out, it's coming up, and it's pretty easy one to remember is the JAK-STAT pathway, and we'll talk about that in a second. So, other family class, so that was our IL-1 family. Now we have our class 1 or hematopoietin family. We'll just call them class 1 family of cytokines, and these are the bulk of these small molecules. They have the effect. They affect many cells, many signal differentiation, activation, antibody secretion. So these are the ones that are talking to T cells and B cells and telling them what to do. They all share these common motifs, and they can be grouped into length. We have our really short helices, IL-2, IL-4, IL-3, and then these larger have long helices, IL-6 and 12. You don't need to memorize that. So they have two different types of protein domains. Again, interesting to know, but I don't really require that you memorize that. And then they have subfamilies as well, these gammas, betas, and GP130s, which are broken down further. And I'm going to zip through those because they're not as important for you to memorize those.
So this is how they work?
Probably not going to be on the exam. So we've got our class 1 family cytokines like IL-2, and most of those have similar receptors. Like the IL-2 receptor is it's going to exist. They're going to have different forms. Again, like I've talked about, we have this low affinity, intermediate, and high affinity form. Basically, what happens is you have this intermediate or this low affinity form that has very low affinity for the IL-2, but then when IL-2 binds to that, then that will cause a cascade that will upregulate the gamma and then the beta, and this will have an intermediate and high affinity form that will really strongly bind to IL-2. And that's pretty common for a lot of these. And then here you have, and here's a different example, for example, receptors for IL-3, IL-5, GM-CSF. They all have their unique alpha that will bind, but then they share a common beta. And in that way, They have similar signals, so they're going to have similar signaling events, but there's this redundancy because you have the same beta, different alphas. The other thing can't remember if they mentioned in the book, but also because they share the same beta, if you have two of them, they don't work synergistically per se because the limiting factor is that beta molecule, and so the beta is competing. They're both competing for the same beta, and so you actually can have some suppressive effects if you have both or trying to get both. So they won't necessarily act synergistically because they're both competing for that same beta molecule. But you can use them almost interchangeably because of that. GP130 receptor subfamily, including IL-6 and IL-12, these are really important because if you knock them out in mice, they don't survive and live, so they are involved in some very important processes in development as well. And that's, and these are important. IL-6 is actually important, and we'll see when we talk about the chapter on maybe inflammation, we'll see that there's this interplay between IL-1 alpha and IL-6 that will cause this inflammatory response, and those are also, again, targets for anti-cancer therapies. So now we're into our interferons. We have type 1 interferons, and a class of those. Again, these have antiviral activity. And then we have a type 2 interferon, which is just interferon gamma. So when people say type 2 interferons, the only one they're talking about is interferon gamma. And these are, as it says, potent modulators of the adaptive immunity. They are basically, if you want to see if there's an immune response, a T-cell response, or B-cell, or anything, usually you're going to look for interferon gamma. So early on they developed ELISAs to find small amounts of interferon gamma, and that will alert you that you are having this immune response. So interferon gamma is one of those that has this pleiotropic effect, and it's really important in signaling like Th1 responses, cellular responses, activating T cells. So receptors for interferons share conserved cysteine residues. They have 12 chains. Again, interesting to read. But here's one of those pathways that everybody has to memorize, and when they're taking immunology, is this JAK-STAT pathway. So interferon, class 2 interferons have, they signal through this receptor that this JAK-STAT pathways, and there's a lot of diversity in this, but they're all the same. So there's different JAKs and STATs. And basically what happens is similar to, again, you have activation of these Janus activated kinases, so they dimerize, and when they bind to their cytokine, and then they phosphorylate signal transducers. So this signal transducer activators of transcription are STATs, so they will phosphorylate the STATs, and then those STATs, once they're phosphorylated, will dimerize, and then go into the nucleus and start transcribing. So this JAK-STAT pathway is important because we have multiple receptors, cytokine receptors for this JAK-STAT pathway. And they all have, not all of them, but a lot of them have different STATs. So if I want to target, for example, say I want to shut down one of these pathways, I can target one of these kinases and specifically target one of these events. So, for example, if I want to have an anti-inflammatory, I might target one JAK or STAT that's involved in inflammation, for example. So So the other family is this TNF family of cytokines, and most of these, they affect development, effector function, homeostasis of cells, so whether cells survive or die. And these are, again, TNF cytokines, maybe they can be soluble, and here they can also be membrane bound, so this is one of the classes that actually have a membrane bound cytokine that a lot of times they can be enzymatically cleaved and then released. So some of the significant membrane-bound ones are listed. CD40 ligand is one of them, Fas ligand, and these can be involved in cell survival or cell death, as in the case of both CD40 and Fas ligand. Their receptors have these cytokine or cysteine-rich domains. You need to know some are cleaved to become soluble variants. Others are anchored in the membranes or by glycolipids. And actually, soluble variants can intercept cytokine before it reaches an actual signal receptor on a cell. And again, that's one of those common tumor cells will often secrete on those receptors to block binding of the ligand in the area. Signaling. This is one of those where these TNFs, the receptors are not fully defined yet, so there's still, we do know that in some cases, they can lead to cell death or survival, and in some cases, the same cytokine can lead to cell death or survival, and through the same receptor. So it's so for example, signaling through the Fas receptor. So we've got our Fas ligand T cells that binds to Fas on a cell that it's going to target for death, and so this is one of the ways that one of the mechanisms by which they believe T cells can specifically kill. So if it binds to a cell and it has its Fas ligand, or the epithelial cell, for example, that's infected with the virus, has this Fas receptor, and it will bind, and then it'll signal through there to basically induce apoptosis. But then we have also signaling through TNF receptors, and they are expressed on the surface of all vertebrate cells. And the life or death outcome depends on basically this race between activation of this caspase 8 and its inhibition by formation of this other protein. So the mechanism's known, but the situation that would lead to one or the other isn't fully understood. And so it could lead to apoptosis or survival. So the same again, cytokine and receptor involved in both processes, depending on some other event, some other deciding event or timing, or maybe how long or how often or how many of them are activated. So this is another area where we're still researching and learning.
We need to resolve it pathway. That's a lot of pathway.
No, you don't have to remember all these. The JAK-STAT pathway I would memorize.
But if you don't remember the pathway, so
If you do remember the pathway, you have better memory than I do.
we need to remember all these receptors, what they do.
It's good to know that, for example, TNF, the TNFs can induce apoptosis. That's an important thing because that's a characteristic of this family, or survival. For example, it's probably good to know that Fas is almost always Fas and Fas ligand are always induce apoptosis, as opposed to, for example, this TNF receptor 1. TNF and different alphas and TNF betas can induce survival or apoptosis. So that's a good thing to know. IL-1 alpha is involved with inflammation, interferon gamma is, just what are the key factors and commonalities between a lot of them.
So it's just a fast fact. It's not a detail.
No, because we're going to get into a lot more detail when we go into specific responses, T-cell responses, T-cell activation, even cell death is part of that process. So we're going to talk about specifics as they apply to T-cells, B-cells, innate immunity.
So this is just introduction, is like a,
Yep.
just know some
Yes.
basics about it.
Yeah. Yeah, you do need to memorize the JAK-STAT pathway, but the others are good to understand, but I'm probably not gonna have you memorizing, what is downstream of what, IL-17, this is our next family of cytokines. Good to associate pro-inflammatory with that, expressed on a variety of cells. The receptors are found in neutrophils, tend to work at the surface of the innate and adaptive immunity. So this is kind of a linker between the two. And there are multiple different IL-17 family members, and they're involved, expressed by, this is a nice review of what cells they're expressed by, their main function, pro-inflammatory, and a lot of these, secreted by neutrophils, induce a Th2 response, or suppress Th17. So here, we see Th2, and that's that humoral response. Th17 is a different, so T helper cells can become these Th17 cells as well, and that's, and I'm not sure if we're going to delve into that as much, but that's another type of responder cell. Again, they have different types of receptors. You don't have to memorize these, composed of 5 protein chains arranged in different ways. They share this fibronectin domain, so this is the commonalities between them all. And signaling through this receptor, again, you might notice that there are common NF-kappa B that was part of our pathway, activation pathway for B and T cells, a couple other, so they activate, results in different, so they have different pathways that they activate. Some of them are common to what we've seen before, and some are new. And then our cytokines is, I'm going to finish up. The cytokines these are small proteins. They're highly conserved, and they have these disulfide bonds that dictate both structure and category. So we've got CX3C, CXC. So we've got different classes that include these different, basically, names. You don't have to memorize those. So you just need to know that the chemokines are going to direct lymphocyte migration. They're G protein coupled receptors. These receptors pass through several times. I'm not going to have you memorize that. And then signaling through chemokine receptors basically just signals for the cell to move to different areas of the body. And so here's the type of signaling event you would see through these chemokine receptors. So there's cytokine antagonists is another kind of thing to keep in mind, that a lot of these cytokines, we have natural antagonists that, like for example with IL-1, we have IL-1 receptor antagonist and inhibits IL-1 receptor activation. We have, there are viruses that have developed a lot of these antagonists, and a lot of these can also induce cancer. So these are kind of things that these viruses have developed to antagonize either the receptor or the antibody itself, and so these are a couple. And we can use these to our advantage, or they can, or the The viruses can use them to their
This
advantage.
one is more effective.
More effective as
Oh,
I don't know what the specificity of any of them are particularly, but they all, and this is how a lot of these viruses, and if you look at these, a lot of these, CMV, the herpes simplex virus, a lot of these are able to escape immune responses pretty well, and this is majorly, and because they've developed strategies to suppress these cytokines. So that shows you how important the cytokines are in the removal of virus, because of those viruses that are more, that tend to be really hang on, are going to have suppressors or antagonists for these cytokines. Inflammatory cytokine levels may be increased in disease state. So in septic, common and potentially lethal. So these, and this is why we have suppressors of these, because they can lead to sepsis. Bacterial toxic shock induced by superantigens. So these are, cytokines can actually be bad. For example, the new cancer treatment with the chimeric antibody receptors, where they put those on T-cells and they attack cells. They actually have to give people chemo, basically knock out immune cells after a while, because they start to really respond so strongly that they could develop sepsis or toxic shock because of the strong immune response. And there are also, a lot of these cytokines are involved in, IL-1 is classically involved in rheumatoid arthritis, type 2 diabetes, and so it's actually IL-1 receptor antagonist is a approved therapy for rheumatoid arthritis, I believe. Also, a lot of these inflammatory cytokines are involved in myeloid cancers, lymphoid myeloid cancers. So they believe that they basically are being hijacked, and they are causing inflammation, and that inflammation actually can be deleterious to immune cells and causes basically restructuring of blood flow to tumors and things like that. So they can be related to disease, or part of causing disease, or hijacked to kind of do that. So cytokine storm is something we may talk about. It's a lot of times, for example, you're not going to die from this influenza. It's not the virus you die from, it's the cytokine storm, the huge amount of cytokines that are secreted by immune cells in response to this virus basically cause such a strong inflammatory response that you get edema, you start to leak into your capillaries, your blood vessels open up to allow, as a response to allow white blood cells to get to the tissues, but you end up basically losing fluids into your lungs and drowning to death. So not really ideal. And then there's evidence of cytokine involvement in, for example, SARS. That's the problem. You're not dying from the virus or the infection per se. You're dying from your response to that. And the good of it is there are cytokine-based therapies for a lot of these. For example, IL-1 alpha or IL-1 betas may be involved in, some of these TNFs are involved in rheumatoid arthritis. And to treat rheumatoid arthritis, we can block some of these cytokines. We can also prevent transplant rejection. IL-2 is hugely important in T-cell activation and T-cell proliferation. So if I have an antibody that blocks the receptor for that, I can block T-cell responses against, or cellular responses against my transplant, or my new liver, or whatever, my new heart. So if we block that immune response. So this is a way of having a specific kind of suppress the immune system, but target the cells that we know are going to attack something. And this is a little more specific, even though it is causing you to be immunosuppressed, it's better than the alternatives. So, summary, I just want you to understand the different families of cytokines. The receptors are important to understanding cell-to-cell communication channels. There are many different types of cytokines in the receptors, making this a complex field, which is why you don't have to memorize it, because we're going to cover everyone in more detail when we talk about T-cells and B-cells. By understanding these molecules, we can better understand disease states. Again, you know, in the earlier days, people thought that certain cytokines actually were beneficial for tumor rejection. Now they're starting to lean in, they're not beneficial. So understanding, and some of these, as you saw, some cytokines can lead to cell death or cell survival. So by better understanding these, we can better understand how to treat different ailments or diseases or defects in our immune response. And so that's why it's good to have kind of a background in that. So that's where we're going to stop. And again, I'll try and get a study guide together for the unit exam 1 week from today. It's going to go through Chapter 5. We're going to cover Chapter 5 on Monday, which is innate immune system. And I'm probably going to have equal numbers of questions for each chapter. Although, if you really want to know what to focus on, I would say Chapter 1 and 2 and 5.
Vai chutar.